Okay then, we can start if you want to start reading.
Excuse me.
Yeah.
Yes. So.
Where were we? Also, I asked to ask. He always knows. I know. We were here. This one. So you can start reading if you want.
A wide range of cyclic RNA species has been found using RNA sequencing. In addition to cyclic protein coding transfers, oscillating long, interchaining non-coding RNA, microRNA, and antisense RNA transfers have been found in the mouse liver. Using global run-over sequencing, PROSEC to
大丈夫? Somebody else can read. If it's not a good time, 大丈夫?
I'm okay.
So it's okay. Don't need to force.
Using global run-on sequencing to identify enhancer RNA transfers, thousands of oscillating enhancers have been found in the place of these cyclic eRNAs correspond with the phases of transcription of nearby genes. Motif analysis of cyclic enhancer at different circadian phases revealed that there was a phase-specific enrichment of transcription factor motifs and the process of motif were enriched at different times. EBOX motif were enriched between Jacob time 6 and ZT9 corresponding to the peak of block demand 1 occupancy. EBOX motif were enriched between ZT9 and ZT15 corresponding to the expression of the EBOX binding protein and the low point of NF-IL3 repressor. RORE and REBDR2 motif were enriched between ZT in Zagreb time 18 and Zagreb time 24, corresponding with the low point of repression of the RORs. The intrinsic transformation specific binding motif were enriched between Zagreb time 0 and Zagreb time 3, which may correspond to GM129, also known as circadian associated transcriptional repressor or chronal repression. Overall, the circadian systemic result agree with the transcriptional readouts accessed by eRNA and gene expression and analysis. The tripartite circadian transcriptional feedback loop, EBOX, BBOX, and RORE drive the majority of circadian gene expression on genome-wide level.
Okay, thank you very much. So, question on this paragraph. No question?
I couldn't understand what the paragraph tells me.
Essentially, what it means is that you can take a look at whatever RNA, whatever population of RNA, you will find rhythms. Okay? So the paper so far has been mentioning about messenger RNA, which is RNA that codes for proteins. But now we are also seeing that other types of RNA, like non-coding RNA, microRNA, antisense RNA, some of them are also found to be oscillating in the liver. Not all of them, but a good proportion of them are oscillating. So this influence of the clock, this the E box, which is the clock and BMAL binding box, the D box, which is DBP and E4BP4, and the ROR-E box, which is the binding site for reverb alpha and ROR. Via these elements, you can find oscillations of many types of RNA, not only protein coding RNA. And in the circadian field, many things were shown to be rhythmic. And if there is a new technique that becomes available, say for example, for the quantification of protein, the quantification of every single protein in the cell, you can be sure that somebody will use that technique and analyze, are there any rhythms? It is okay, but the problem of this kind of discovery is that if you measure many things, you will find some oscillations. So what does it mean? What is the physiological function of these oscillations? Why are some long non-coding RNA oscillating? It's not known. It's just showing that there are rhythms, but it doesn't provide any answer as to why these rhythms are there. Which is the problem, I think, of these discoveries. And I am not so interested in doing this type of paper. You can check all the papers he said, and we are going to see about that now, binding of clock, binding of BMAL. Yeah, it's rhythmic, but what does it mean for our health? It's often missing. So they show that there are rhythms, and then that's good, but it's not really kind of a new concept. We know now that rhythms are everywhere. So whatever, you can look at whatever, you will find some rhythms. Okay. Any other questions for this? Zeitgeber, do you understand? Zeitgeber. ZT. Do you remember what is? It's German, Zeitgeber. It's German word. It's written on the side name. A standard of time based on the period of an environmental synchronizer, or Zeitgeber. Zeitgeber in German means time giver. Okay. All right. Then let's go to the next paragraph. Okay.
Using whole transcriptome RNA-seq or nascent-seq combined with mRNA-seq, two divergent sets of cycling genes have been found. Cycling nascent transcripts and cycling mRNA levels. Only about 20% of cycling mRNAs also had a corresponding cycling nascent transcripts, indicating that other RNA processes are likely to be contributing to steady-state circadian mRNA expression. Several different classes of cycling transcripts have been observed, including the following: first, cycling transcription and cycling steady-state mRNA. Second, cycling transcription and no cycling mRNA. And third, no detectable cycling transcription, but cycling mRNA levels. The first class is typical for the core circadian genes and their high amplitude direct targets. The second class is typically seen in highly expressed genes in the liver, that have mRNAs with long half-lives, such as albumin. The third class represents transcripts that could become rhythmic via circadian variation in mRNA turnover, RNA splicing, polyadenylation, microRNA regulation, and other post-transcriptional steps. Whether rhythmic translation or protein levels ultimately occur has also been revealing. For example, rhythmic diurnal polyadenylation occurs in 2.3% of expressed genes in the liver, and 1/5 of these genes, third class, lead to rhythmic protein expression that correlate with poly-A tail length in the absence of rhythmic mRNA levels. Ribosomal profiling has also revealed substantial numbers of examples of circadian translation in U2OS cells and liver cells. Interestingly, there are at least 150 examples of translationally driven oscillations in the liver for which no mRNA rhythms are present. Thus, both transcriptional and post-transcriptional regulation are critical for circadian gene expression.
Okay. What about this paragraph? What about this paragraph? 174. Any questions about this one? Maybe they are. You should not hesitate to ask questions, okay? Especially on methods, because using whole transcriptome RNA-seq or nascent-seq combined with mRNA-seq, is it understood really? I'm wondering. Nascent-seq. Don't hesitate. Steady state mRNA. Cycling transcription and low cycling mRNA. These things, you understand what it means?
No.
Just like the cycling transcription, cycling steady states, mRNA. Different scenarios.
Yeah. So if you draw let's draw the most simple case that you see in textbook, where you have DNA, you have, that's the poly-A tail of the messenger RNA, you have mRNA, and you have proteins. And what has been the first thing that has been done, because the techniques were available quite early, is that people started to look at, what are there rhythms at the level of RNA, messenger RNA? So they extract RNA from whatever samples, and we quantify how much RNA there is for each gene. That's okay. Then you can do the same with proteins. You extract all the proteins and you measure how many proteins are oscillating. But if you do that, this is the steady state RNA level. This is the steady state protein level. At one time, how many RNA you have of each gene? It's not dynamic. You don't know how many RNAs are being made. You only know how many mRNA have been made, are currently existing in the cell. This is steady state. And you see rhythms. You see quite a lot of rhythms. But now, if you don't take a look at the amount of RNA that is currently in the cell, you are looking at what is the amount of RNA that is being synthesized. Where you have a polymerase here that is actively synthesizing a specific mRNA. Then you can take a look at that and see if the RNAs that are currently being made, and you do the same thing at different time points, and you want to know, is the active synthesis of RNA changing with time? And the answer is yes. If you check, this is time, and this is the synthesis of RNA, then for some messenger RNA, you will find that they have a rhythm of synthesis. Okay? So the synthesis of some mRNA has a circadian rhythm. But if you take a look at this and you take a look at that, you can find the same mRNA, of course, but only 20% will have a rhythm at the level of the synthesis of RNA. When it's still linked to the RNA, it's being synthesized by the RNA polymerase. RNA are rhythmic, but only 20% of these ones. Only 20% of these ones. Which means that there are a lot of steps here that leads to the fully synthesized steady-state messenger RNA. And these steps, in addition to this one, which is like clock and BMAL mediated, activation and everything, these steps might also generate rhythm. 80% of the rhythm of the mRNA is not understood. This is what it means. 20% of the rhythm arise at this level, the new synthesis of RNA. But mostly, 80% of the rhythmic gene, the rhythm comes from somewhere else. And there are many different ways. What may be the different steps? Can you tell me what might be the different steps by which rhythm in steady-state mRNA might be generated, other than nascent synthesis? What might be other steps? Polyadenylation, yes. So, synthesizing the polyadenylation. This is important to make a mature messenger RNA. It's what is the length of the polyadenylated tail? When is it made? This might be one of the possibilities. What else? Splicing, that's a very good one. So, what is missing here is that first you make mRNA that still is having exon and intron. And then after that, the the introns are being spliced out and only the exons remain to make the full messenger RNA. And this splicing may also be regulated by the clock. So transcription is not rhythmic, but there is a certain amount of pre-mRNA. This is called pre-major messenger RNA. But then at different times it will be spliced, and at sometimes it might not be spliced, and it's kept as a storage. So splicing is one. Polyetyl synthesis, it's one. What else?
RNA turnover.
RNA turnover. Yeah. So if the degradation of RNA is controlled by the clock, the steady-state messenger RNA. So the synthesis of new RNA will not change, but if the synthesis is constant and only at certain time you actively degrade RNA, then this will also generate rhythm at the steady-state mRNA level. So this is the turnover. Turnover. What else? What other mechanisms could generate rhythm in messenger RNA?
Transport.
Yes, that's a good one as well. That's a good one as well. Maybe the turnover of RNA is mainly in the cytoplasm, but messenger RNA is made into the nucleus. This is the nucleus, this is the cytoplasm. If you have a constant turnover, but only at certain time the RNA is leaving the nucleus, this could also generate rhythm, indeed. So this is my transport. So the point is, many steps may also generate some rhythms. And the mechanism by which 80% of the RNA are rhythmic, but we don't know where the rhythm comes from. We don't know. We still don't. 2.5%, according to the data, only 2.5% of RNA, they are rhythmic at the poly A. So it's a very small fraction of the genes. So is it biologically significant? So there are many things we don't know. So not everything, even though what I told you before is the clock really operates at the level of gene expression, at the level of transcription, so clock and BMAL bind to the DNA and activate RNA polymerase to synthesize. This is maybe apparently quite a small fraction of RNA that are rhythmic. So still many things are not known. My research here actually works on this level. What happens between RNA synthesis and protein levels? Okay, is that okay?
Okay.
All right, then maybe we can continue.
Genome-wide circadian regulation of transcription RNA-B2 and chromatin states. In the mouse liver, cycling pre-mRNA levels using the intron RNA signal were coordinately expressed in time, with a peak at CT15. Thus, there is a peak of globally coordinated circadian transcription that occurs at the beginning of the night, during the active or awake portion of the mouse circadian rhythm, which is not seen at the mRNA level, which is a bimodal pattern. To further analyze the global rhythms in nascent transcription, RNA polymerase II occupancy has been measured during the circadian cycle. The large subunit of RNA polymerase II, POLR2A, contains a carboxyl terminal domain that is post-translationally modified at different stages of transcription. RNA polymerase II is recruited into the pre-initiation complex in the hypophosphorylated state, recognized by the 8WG16 antibody. Surprisingly, RNA polymerase II 8WG16 occupancy is highly circadian in the mouse liver, with a peak that correlated with the intron RNA peak. The first step in the initiation of RNA polymerase II involves phosphorylation on serine 5 of the CTD of RNA polymerase II. RNA polymerase II serine 5P occupancy is also circadian, indicating that both recruitment and initiation of RNA polymerase II are under circadian control. An antibody against the RPB2 subunit of RNA polymerase II revealed that RNA polymerase II occupancy for promoters and gene bodies is rhythmic and co-varies over time. Rhythmic loading of RNA polymerase II to promoters rather than a rhythmic transition from a pause state to productive elongation accounts for the majority of rhythmic transcription in mouse liver.
Okay. What about this one? I actually have a question. Why are they talking about antibodies? Why in this paragraph they are talking about antibodies? That's just to see if what we saw in the previous lecture is remembered. Why are we talking about antibodies to RNA polymerase II? Why are we talking about antibodies? I thought we were talking about RNA and expression, and why are we not talking about antibodies? RNAP2, so RNase polymerase II, is recruited into the pre-initiation complex in a hypophosphorylated state, recognized by the 8WG16 antibody. Why are they talking about it?
Because they want to measure the rate. They want to measure how much is the gene is transferred. It's transcribing the amount of transcription.
Yes. Although in this case, they are not measuring transcription. They are measuring something else. Yeah, of course, via this technique, they know that the RNA polymerase is making RNA at a specific location. That's true. So they are, although they are not looking really at RNA, they are looking at the activity of the RNA polymerase. But why do they need an antibody?
Because it recognizes how much is the RNA polymerase complex is phosphorylated.
Yes. They have an antibody against a hypophosphorylated state of the RNase polymerase. This hypophosphorylated state is a marker of only the recruitment of RNA polymerase. The RNA polymerase is there, but it's not active. It has just been recruited. It's not doing anything, but it's there. But my question remains, why do they need an antibody? What are they measuring here? What are they measuring here that they need an antibody for? It's very similar to maybe one or two lectures in the past.
RNA polymerase II is somehow not make covalent bond with DNA by chipset.
ちょっと違う。ちょっと違う。Yeah, RNA polymerase will not really make any bond with DNA. It will sort of take DNA and start to synthesize RNA. But first, it must be recruited at a specific site. It's there, and then when it is activated, it will start making RNA. But why do they need an antibody? What do they do? What do they do in what they explain there?
They want to separate the recruitment and the activation of RNA polymerase.
Yes, exactly. They want to separate RNA polymerase that is recruited but doesn't do anything, and RNA polymerase that has been activated after recruitment. So then they have two antibodies, one that recognizes the recruited state of the RNA polymerase, and one antibody that recognizes the activated state, because RNA polymerase has been phosphorylated. And they have an antibody that recognizes this phosphorylation of this RNA polymerase. But what do they do? What kind of techniques do they use to know, okay, this RNA recruitment of RNA polymerase at this gene, at this gene, at this gene, at this gene has a circadian oscillations. Activation of RNA polymerase at this genomic location, at this genomic location, at this genomic location has a circadian oscillation. So what do they do? We have seen something very similar in the previous lecture. What do they do with the antibody? Using the antibody, they could simply extract protein. Okay, they could extract protein and use these antibodies to quantify how many of the recruited RNA polymerase and how many of the activated RNA polymerase there is in the cell. They could do that. They could check the amount. But in this case, this is not what they are doing. In this case, and the important word is occupancy. This is the key word. They are not checking the amount of the protein, really. They are checking at the level of occupancy. What does it mean, the occupancy level? Occupancy is something that means occupied. We are now currently occupying this room. Okay? This is what it means. Occupancy, this is what it means. They are checking the occupancy of RNA polymerase, recruited and activated states, using an antibody. How do they do it? Try to remember what we saw last time with clock and VMAD and, like the previous figure. They have done the same thing, but using different targets. They weren't using polymerase, they were using clock and VMAD. So what were they doing? They previously measured the occupancy of clock and VMAD. Do you remember how they were doing it? You can try to go back in the paper and try to see the answer. How was it? You know, this is figure, this is figure. Figure on page 172, the figure A. This is what they were doing with clock and BMAL and other proteins. And even a figure before that as well. Why do I have the same copy of the same page? So anyway, 27 is 127. Okay. Page 3, 4. The figure 3A is also the same. They were doing the same thing. But here it's not with clock BMAL per crime, it's with RNA polymerase 2. What was it? Do you remember? Try to look at the paper, try to understand what they were doing, check the legend of figure 3, check the text, and tell me if you understand. So they are not measuring the amount of RNA. They are not measuring RNA. They are measuring occupancy of proteins. Occupancy of what? What do they occupy? We are occupying this room. But in this case, RNA polymerase 2, what does it occupy? What kind of? Try to read. It is difficult. It is difficult because it's not a technique that is that mentioned in many lectures. Now you can find the same word in the figure 3, in the legend of the figure 3. They also talk about occupancy. First of all, I guess, occupancy of what? Occupancy not of promoter. Occupancy at promoters, right? Occupancy at promoters. So, occupancy of clock and BMAL at promoters. Or in promoters, yes. Occupancy of clock and BMAL at promoters. Okay, so one by one. So we have gene X. Maybe this gene X is per 1, which we know it's a clock and BMAL regulated gene, and they are measuring occupancy of clock and BMAL at the per promoter. So this is the gene, where this is the coding sequence, and here this is the promoter, right? That's the promoter. That's the promoter. And at certain region of the promoter, sometime clock and BMAL is bound. So the occupancy of clock BMAL at the per 1 promoter has a circadian variation. Sometimes it is not there, sometimes it is there. And you can see the figure 6 that is just on top of what we are reading. You have the x-axis shows circadian time from 0 to 24, and then you have the green and the mauve color, BMAL1 and clock. And this shows the occupancy. Okay. This shows the occupancy of BMAL clock at the promoters of many genes. Okay, this is the total occupancy, not only of one gene, but of total. And you can see that the maximum occupancy of BMAL1 is maybe 6.1, circadian time 6.1. And clock, it's not so different, 7.3. Okay, fine, but why do they need an antibody for that? So what they are doing now, it's not clock and BMAL occupancy, it's RNase polymerase. RNA polymerase 2. They are now checking the occupancy of RNA polymerase at site of transcription. Either the active, so the one who is going to start actually making RNA, and the kind of a recruited one, RNA pol 2. This is the recruited one, and this one is the active. So they have two antibodies, but why is it needed? Why do they need an antibody for that? Why do they need an antibody to measure the occupancy of clock, BMAL, or RNA polymerase? Okay, I will distract you. First, a simple question. RNA polymerase 2. It implies that there is RNA polymerase 1, right? If it's RNA polymerase 2. How many RNA polymerase our cells have? 3. Yeah, we have 3. Why we have 3?
Link 2 is involved in most genes transcription.
Yeah, so polymerase 2, this is the polymerase that is making messenger RNA. Polymerase 1, it's a polymerase that makes different types of RNA. And polymerase 3, I think it's making ribosome or something. I don't remember which is which, but there are actually 3 RNA polymerase. The one that they are interested in is polymerase 2, the one that is making messenger RNA. So, why do they need antibodies?
To separate polymerase 2 from other 1 or 3?
Yes, of course, yes. But they want RNA polymerase 2, so they have antibodies against RNA polymerase 2. But how will this give a measure of the occupancy of RNA polymerase 2 at promoters? Yeah, they could do the same experiment with an antibody against RNA polymerase 1, and then they will check, is there any circadian rhythms of occupancy of RNA polymerase 1? But how do they measure occupancy with this? And they are quantified, right? They are quantified. If you take a look at the y-axis of the figure 6, they say percent of peaks. Percent of peaks. So and if you take a look at the figure 3a, the y-axis, they are not saying it, but it's also, you have like peaks, right? It looks like peaks in the per1 promoter. The occupancy of BMAL1 and clock, it's kind of, you have peaks. You don't remember? Don't remember? What are they measuring?
The frequency of the specific reads. Like
The CS, this is very close. They are looking at reads. They are looking at reads of what? Reads of what? Reads. Read. They are reading something. They are reading something and they count how many of these things they are reading. Okay? So for example, if I write the word wow, there are 4 reads of wow. This is what they are counting. 1, 2, 3, 4. There are 4 reads of wow. This is what they are doing when they say reads. But of course, they are not quantifying wow. They are quantifying something. Reads of what are they measuring? Reads mean copies, right? 4 copies of wow. Yes? No? So they are measuring reads. But reads of what? Let's take 2 time points. If I use the figure 6, and if we look at 2 time points along the x-axis, we look at 7, CT7, and you can see that the BMAL1 peak is the highest at CT7. And if you take a look at time 18, then you don't have BMAL1 there, right? This is understood? You can see? Yeah? Okay, so let's see. CT6, this is the first. And we have CT18. So CT6, this is high binding, this is high clock and BMAL occupancy, and CT18, it's low BMAL1 occupancy. And you know that they have what they want. They want BMAL1. This is one of their tools. It's the BMAL1 protein. And they have an antibody you can exchange BMAL1 for polymerase 2. It's exactly the same technique, okay? It's just different antibody. And they have an antibody against BMAL1. And promoter, what is promoter? What is promoter? What kind of biological molecule is a promoter? This is a very simple question.
Sequence.
No, sequence is not a molecule.
DNA.
What? DNA, yes. So they have DNA. So you are right. If they have DNA, they have sequence. They have reads. Okay? So there is a BMAL1 protein, there is an anti-BMAL1 antibody, and there is a DNA that corresponds to the promoter. So now we need to assemble these things together in CT18 and CT6. Is that a hint? Is that a hint? Let's take the promoter of DNA. The DNA component is promoter of a given gene. Let's say, again, this is the PER1, but it can be anything. So, of course, the promoter is DNA. It's also there at CT18. That's just the same. It's always there. That's PER1. And here, this region is the promoter. Okay? From the figure, we can understand that at CT6, BMAL1, which is the protein component, is occupying the promoter of PER1. But at CT18, it is not. Okay? So, from there, how can we quantify? You know that there it's bound. We sort of reverse engineer the result. This is bound here, and this is not bound. How could you quantify that? Using an antibody. Okay, first of all, you don't have only one promoter. You have many promoters, because what you are doing, it's extracting RNA, extracting DNA from a lot of cells. Okay, a lot of cells. Each of these cells will actually give you 2 copies of each promoter. 2 copies of per gene, right? So I will draw here many copies of the same gene because you have extracted many cells. And here you have, if you have done your experiment correctly, ideally you have the same amount of copies. So you have the same amount of starting cells. But in here, I told you, BMAL1 is not occupying the PER promoter, but because we are dealing with biological samples, sometime randomly you may have BMAL1. So let's do this. In this case, BMAL1 is always occupying the PER promoter. All the copies of DNA that you get, they have BMAL1. But in this case, maybe only one of these copies has a BMAL1 weakly attached to it. Because, there is some random variation. Okay. So then what do?
You use an antibody to precipitate the BMAL complex, and you cut the DNA so that and then you actually basically the idea is that you only keep those V's that's attached to BMAL1 and count the
Yeah.
frequency of the V's.
Exactly. So the first thing you want to do is that in the cell, BMAL1 will be associated with the DNA. It will be bound to the DNA. But first, you must make a covalent bond between DNA and BMAL1. So you want to make the association more stronger than it is in the cell, because you are going to do a lot of things to your sample. So you make a big link, and of course, here's one. That's the first step. You must cross-link. Then what you are going to do is digest your DNA, so that you are making random fragments. You cannot control where the break is going to happen. You are going to cut your DNA into small pieces, and you are going to do it here as well. I feel like a mad guy. You are going to break your DNA into small pieces. Okay. The key you can see here is when BMAL1 is bound, you don't cut the DNA here, because it has been stabilized by BMAL1. So you never cut in between, or very rarely. And then, using the third thing, using your antibody, you are going to bind with your antibody BMAL1. You are going to bind BMAL1 with your antibody. This antibody is very heavy because it has been bound to a very heavy plastic bead, or it could be a magnetic bead, Jicky bead. So you can purify this antibody bound to BMAL1. And because you have an antibody that is heavy and that is specific against BMAL1, you can remove from these big DNA samples only the fragments of DNA that are bound to BMAL1. So, you are essentially removing all the DNA that is not bound to BMAL1. I do this one. So then that will be nicely. There you go. And of course, here there is no BMAL1, so you don't capture the fragment of DNA bound to BMAL1. Only you get one fragment. And then what do you do? The last step of this experiment, what do you do? You
just use protease to
yes. Then next step is that you destroy BMAL1 along with the antibody. You destroy BMAL1 here. There you go. And then what do you do?
sequence and count.
Yeah. So you actually, they are very big machines. They are quite small, but I mean big in terms of capacity, who can sequence every single bit of DNA you have in your sample. So actually you will know that this sequence and 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 this sequence is exactly the same. And it's a sequence that contains the E-box CACGTG of the PER1 promoter. So you are going to count, okay, how many copies, how many reads of that sequence I have. 1, 2, 3, 4, 5, 6, 7. I have 7 reads. Normally reads, they are thousands, not only 7. Okay? And here you have 1. So then if you plot time, CT6 and CT18. You have reads. Then you get a high amount of reads for CT6, and you have very small amount of reads for CT18. And then you say that the occupancy of BMAL1 at CT6 at the per 1 promoter is higher than at CT18. This is what it means. Okay? This is what it means. And because you can analyze and quantify every single sequence you have in your sample, then you can do this data not only for the per 1 gene, but you can do it for every single gene in the genome that is occupied by BMAL1. And this is how they make the figure 6. And they have done exactly the same thing for the RNA polymerase 2. Using a different antibody, they have taken the DNA fragments that were associated with RNA polymerase and so on and so forth. This is what they are doing. Is it clearer? Yes? So they just have a very nice techniques and they use it with many different proteins. BMAL1, NPAS2, the previous figures we have seen. But now they are just checking RNA polymerase occupancy. Any other thing from this paragraph? It's kind of funny, right? They say in the mouse liver, cyclin pre-mRNA level were coordinately expressed in time with a peak at CT50. And then a little bit later, they say, thus, there is a peak of globally coordinated circadian transcription that occurs at the beginning of the night during the active or awake portion of the mouse circadian rhythm, which is not seen at the mRNA level, which has a bimodal pattern. What does it mean when they say that the mRNA level has a bimodal pattern? There are two peaks, yeah. So what they say here, let's draw a graph, is that if they check pre-mRNA level, so this is time here, time of day, and this is just total pre-mRNA level. You have this is RNA level. This is RNA level. And in red, it is total pre-mRNA cycling level. Only one peak. One peak of pre-mRNA, but they have two peaks of messenger RNA. A bimodal pattern means two peaks. So here they are discussing about the overall pattern. So if they calculate the average between all the cycling genes, all of them, they see that the average is always high at a specific time for pre-mRNA, but it is a bimodal pattern for mRNA. Can you explain that? They are calculating the average or the sum of all the rhythmic genes. So why do they have a bimodal pattern for mRNA and they have a single peak? it may not be, the phase may not be correct, right? But the meaning of what they say is like this.
Some of the oscillation pre-mRNA effects cancel out, so
some of the oscillations may cancel out. Yeah, that's possible. That's possible. So they are discussing here about the fact that the peak in pre-mRNA is during the active phase of the mouse. So this is the dark phase, right, during the night. But the peak in mRNA level can be during the day or during the night. This is what it means when they say bimodal. Okay? The peak of pre-mRNA is only during the active phase, but the mRNA peak is during the day or during the night. So let's say this is the day here, and this is the night. So the peak in pre-mRNA is mostly during the night. But there is a peak during the day for messenger RNA, and there is a peak during the night for messenger RNA as well. So what does it mean that? How can there be a bimodal pattern? So keep in mind that this is not one single gene they are looking at. Okay? Now they are looking at mRNA. They are not looking at anymore at binding, this paragraph, this specific paragraph. It's about RNA. Okay? It's intronic, pre-mRNA by measuring the intron, or major messenger RNA. So it's a different pattern. Why it is possible? Where does this bimodal pattern come from? One peak during the day and one peak at night.
Is that free translation or modification?
Posts?
Post translation of no?
No. It's more simple than that. It's more simple than that. They have taken all the genes and all the rhythmic gene, and they have calculated, well, they have kind of superposed each other, all of them together. And they see that for all the rhythmic pre-mRNA, there is a single peak. And for all the rhythmic messenger RNA, there are two peaks. What does it mean? What it means, it's simply that every single transcript, every single pre-mRNA has a very similar peak, like this. So when you add them together, they can be a little bit different, but all of them really have one peak. But messenger RNA, as we know now, transcription, there are many different levels of transcription. There is a BD-box, there is E-box, there is RORB. So messenger RNA have very different pattern of expression. Some messenger RNA have this kind of expression. Some messenger RNA have this kind of expression. Some messenger RNA have this kind of expression. And so if you make an aggregate of all these expressions together, you have that some of the genes are high during the day and some of the genes are high during the night. Okay, this is what it means. This is what it means. It's bimodal because there are different phases of RNA. Okay, there are some genes that are high during the night, some genes are high, some mRNA are high during the night, some mRNA are high during the day. But if you check pre-mRNA, which is really a measure of this really transcription, not all the RNA processing business, there is one single peak. You're all sleeping, or am I making
No,
sense?
but I mean, if like what you're saying is that we have two peaks because some of them are so like express a day but not express a night, and some of them are exactly the opposite. So I have the basically two peaks. But if it's that, in that case, won't the effect cancel out because some of them have so low expression at night, some of them have so high expression. So it will be, I mean, at least the peak will not be as obvious as the pre-mRNA, because in the pre-mRNA case, everything is concentrated at the same time. But even if you have two peaks for mRNA, it will be.
Yeah, they will cancel each other. They will cancel each other, especially if they calculate the average. If they calculate the average, you are right. So if all the pre-mRNA peaks are really within a few hours of each other, and if you calculate the average, then you have a huge peak. But if you calculate messenger RNA, and if you average all of them together, then what you may get is some sort of thing like this. And it depends what kind of mathematical thing they do. I think what they are doing, they are just superposing all of them together. Like, for example, they take genes that are expressed during the day, and they see that there is a peak here, and they add them together. They don't make the average, they add the data together. And then they have genes that are expressed during the night. And when you add them together, then you have this. If you calculate the average, you get something like this. Or even something like that.
Yeah, that's what I saw. I think there shouldn't be even peaks like
average. So this means that although there are different level of regulation, there is maybe a lot of things happening here that actually generates two phases. There are actually more than two phases, but mainly two phases. But this single peak of intron means something. So we can continue the paragraph. So then they decided to further analyze the global rhythms in nascent transcription. So do you understand the link between nascent transcription and intron? What is the link between nascent, so they are talking about intron, right? Pre-mRNA level, so they are quantifying messenger RNA that contains intron. So this is not major RNA, this is pre-mRNA. And then they say to further analyze the global rhythms in nascent transcription. And then they speak about polymerase II. So what is the link between nascent and pre-mRNA? What is the link between quantifying pre-mRNA levels and quantifying nascent transcript level?
Pre-mRNA level and nascent transcription level has the same kind of oscillation.
Yeah, it's very similar, right? It's very similar because you have DNA here, and then you have an RNA, a nascent RNA. Nascent means just being born. This is what nascent means. You have a messenger RNA that is just being synthesized, and of course, it is still containing the intron. So when you quantify the amount of intron containing mRNA, it's not the same, but you are very close to quantifying nascent RNA synthesis. It is very different when you quantify an RNA that has a poly-A tail and has been spliced and everything. So this is very different from the nascent here. But intron is not so far. Intron is not exactly the same, but it's not so far. So this is why they say, okay, very interestingly, when we check the rhythm of this against the rhythm of that, the pattern is very different. So the difference may originate in when the mRNA is being synthesized. So let's just check that by lesson sec. Okay. And then we will finish this. Paragraph here. So large subunit of RNA. Well, this is just it's a big protein that has two different subunits. And then you can see from the figure 6 that the red peak, this is the serine 5 phosphorylated RNA polymerase 2. This is in the recruited stage. And then you can see that later at CT12, CT14, then the 8WG16 hypophosphorylated state just before starting the transcription, it's different. So it's really controlled at two different times by the clock. Both the recruitment and the initiation of transcription. And then you see the last part of the paragraph, an antibody against the RBP2 subunit of RNA polymerase 2 revealed that RNAP2 occupancy for promoters and gene bodies is rhythmic and covaries over time. Rhythmic loading of RNAP2 to promoters rather than a rhythmic transition from a pause state to productive non-degradation account for the majority of rhythmic transcription in the mouse. So again, this is very interesting for circadian biologists. That is very nice. It's really not only there are transcription factor that regulates rhythmic transcription, but really globally, RNA polymerase 2 is recruited and activated with a circadian rhythm. So what is the question you want to ask? What is the question? This is the last thing we see. If you read like this, okay, fine, so there is a mechanism by which clock and BMAL regulates transcription, but it seems there is a different mechanism in which polymerase is recruited at a specific time that does not seem to depend on clock. It's very strange. What kind of question you want to ask when you read such paragraph? You see, like you have a peak for BMAL1 and you have a peak for clock. They're kind of synchronized together. But you have a peak for Cry2 and you have a peak for PER1. And you have also, they don't show it here, but there is a peak for DBP probably, and there is a peak for reverb alpha probably, which will be different. But if you take a look at the peak for RNA polymerase recruitment, there's only one single peak. So these RNA that are rhythmically controlled by rhythmic recruitment of polymerase 2, they have only one peak. One single peak. So what is the question you want to ask? As to why that is so. Not maybe molecular level, but at the body level, at the physiological level. What do you want to know when you see something like this?
The peak of the RNA polymerase 2 is one peak because it has many for different genes, they have different peaks, but when you add off, that it becomes one peak, or it's only one peak for every gene.
So maybe there may be smaller peaks, similar to maybe what you say, is that maybe when you calculate so large level of things, so peaks cancel each other, and actually you may have different peaks. But I don't think so. There might be slightly different peaks, but not antiphasic. Certainly not antiphasic. So it seems that there might be very small peaks somewhere else, but maybe the biggest peak, the most significant one, is really, as we can see, CT0, CT24. This is really the biggest peak. What is a question you might have looking at this data?
The only one peaks of RNA polymerase II can make other genes have so many peaks, so many different peaks.
So you mean that one single peak of RNA polymerase II somehow may trigger different peaks of other things by interactions? That's always possible. That's always possible. We may well find out that sometime actually maybe the serine phosphorylation, the serine 5 phosphorylation may not in all cases mean the same thing. There are always things that we don't know. That's possible. But let's say it is true. Let's say what they show is really that there is one single peak of RNA polymerase II recruitment. And that drives rhythmic expression of many genes. So let's say this is true. Let's assume this is true. Then what do you want to know? What do you want to know about these genes? So what they don't say is that the genes that have a high occupancy of RNA polymerase in their promoter, are they the same one that have a high occupancy of BMAL1 and clock? Are they the same? We don't know. We don't know whether they are the same. We know that they are genes that have a high binding of RNA polymerase in their promoter. But if we try to see the genes that are regulated by clock and BMAL, the genes that are regulated by RNA polymerase, are they the same? Or is it it seems to be a subfraction, just like there are genes that are regulated by polyadenylation tail at the level of mRNA. Maybe there are genes that are regulated by RNA polymerase II recruitment only, and there are genes that are regulated by clock and BMAL. Are they the same genes? We don't know yet from this paper. What is the physiological function of controlling genes with clock and BMAL and controlling genes with rhythmic recruitment of polymerase II? Or is it linked, as you suggested? So this is the kind of things I want to know when I see this kind of data. Okay, are these genes the same? Are these genes the same or not? If they are not the same, maybe they have a very specific function. Maybe the genes that are controlled by rhythmic recruitment of RNA polymerase II, as they say, it seems to be something for the night, so maybe this is needed for the active phase of the mouse. We don't know. But this is the kind of things I want to know. Why so many different rhythmic input into gene expression? What is the meaning of that? This is what I want. Okay. It's 6 o'clock. If you have any question, don't hesitate. Well, I'm always, when I read a paper saying we have found 2% of genes that are rhythmic at the polyadenylation level, I find it very strange because statistical significance is defined as 5%, right? So when you say that 2% of genes, how strong is that? How significant is that? Of course, if you measure every time and you always have the same genes, then it's pretty strong. But I am not so sure. And so this level of the polyadenylation tail, it's mama. Let's say it has not been confirmed very much. Of course, it's from his team. This is from his team. This is a lady called Carla Green who has shown this polyadenylation thing, but nobody else has confirmed that yet. So I'm not sure how significant it is. Clock and BMAL and transcription, this is very strong. It's extremely strong. Many people have shown the same thing. But all these, like 2% of genes controlled by the 2% of controlled by that, that's a little bit less. So take it with a pinch of salt. As far as I'm concerned, the most really significant input to the clock is really the clock and BMAL binding to the box, regulating per 1, per 2 reverb alpha. This is very, very strong, very solid. All the other ones, they may be true, but they await for demonstration of their importance, I think. Okay.